Roots are essential for plant growth, but their effectiveness is greatly enhanced by symbiotic organisms. This can help the plants to access nutrients and to defend themselves against pathogens. Indeed, with symbiotic organisms, plants grow much more vigorously, as we can see in these maize plants. The more symbiotic organisms, the better the plant will grow. By measuring how many of these organisms are present, we can learn to understand how they respond to factors such as agricultural practices, soil conditions, or plant genetics. Until now, quantification of these symbiotic organisms has relied on microscopy, which involves staining the roots and meticulous observation. This is time-consuming and subjective because it depends on the observer's level of expertise. Instead, researchers now can use a method called quantitative PCR or qPCR. This technique measures the DNA of the organisms, making it possible to estimate their numbers in soil or roots with much greater speed and accuracy than traditional microscopy. Do you remember those nose swabs during the COVID-19 pandemic? Sometimes we did a quick test at home, but the more accurate ones in clinics were analyzed in a lab using qPCR. qPCR works by detecting genetic material in a sample. Even if there is only a tiny amount, the technique makes enough copies to measure it. That's why it was so good at detecting the virus, even in the early stages of infection. Also, labs can process hundreds of samples at once. But qPCR is not just useful for detecting pathogens and finding diseases. In our research, we use it to look at the beneficial microorganisms living in plant roots. Instead of asking, is the virus present, we ask, how much of this beneficial organism is present? Absolute qPCR works by comparing the amount of DNA in our sample to a reference scale from samples with known DNA amounts. It's like having a chart that shows what a strong, medium, or weak signal should look like. Then we test the root samples, we can see exactly where they fit on that scale. There are 6 steps in the qPCR process. Step 1. Collecting the sample. Just like a COVID swab, we start with a bit of root or soil. Step 2. Extracting the DNA. The genetic material of all the organisms is pulled out of the sample. Step 3. Preparing standards. Scientists prepare a reference sample with known DNA amounts, like having a ruler to measure against. Step 4. PCR cycles. The DNA is copied again and again in a machine, so even tiny amounts become visible. Step 5. Fluorescence detection. A dye glows when it binds to DNA, and the machine measures the brightness after each cycle. The brighter the sample, the more DNA is present. Step 6. Comparison. By comparing the glow from the root sample to the references, we can see how much of the organism's DNA was there in the first place. We have now quantified how much of a particular DNA sequence was in our initial sample. For human health, that tells us whether someone is infected. For plants, it tells us how many beneficial organisms are present in their roots, something we could never measure as precisely under the microscope. If you'd like to learn more about qPCR and how to quantify symbiotic organisms, check out our other video. You can also find more details in our publications or explore the latest findings from the Root to Rest project on our website. And if you'd like to follow our journey, don't forget to subscribe to our channel.